Hi everyone, welcome to Lecture 8G of Useful Genetics, where we're going to be using crosses to find out the locations of autosomal genes relative to each other. We'll talk about scoring the effects of crossing over when it's hard, and about a technique called a test cross that makes it all much easier. And we'll use this to solve a particular genetics problem. So this is a reminder diagram of crossing over from Module 7. And one of the things that geneticists use crossing over to find out, in addition to using it as a tool for understanding how meiosis works, geneticists use crossing over to find out where the genes are in the genome, which genes are on the same chromosome, and how the genes that are on the same chromosome are arranged. And the first step is simply to estimate the distance that separates two genes. For instance, the big distance there, short distance there. And we're going to do this in the context of a chicken problem. So we're starting with the F1 progeny of a cross between chickens that were homozygous for recessive alleles of two different genes, C and D. So some of our chickens had the big C allele, homozygous, they were black. Some had little c, they were gray. Some had big D homozygous, they were shiny. Some had little d, their feathers were dull. What we want to answer is whether the C and D genes are linked. Now, I'm not telling you the genotypes of the parents. I'll tell you the genotype, though, of the F1, which you, of course, could work out here. Must have been big C, big D, little d. We want to find out if the C and D genes are linked. Now, you might think that the easiest way to do this would be to just cross these F1 chickens, these are F1s, to get an F2. That's the F2 refers to the offspring that you get when you cross the F1 birds with each other. But a minute's thought makes you think that this is going to be a hard problem. You might say, well, even if magically you knew the genotypes of all the chickens in your F2, it would still be hard to figure out how much crossing over had happened, because crossing over had happened in both of the parents. And if you didn't know the genotypes, all you knew was the phenotypes, it would be a lot worse, because you'd also have the problem of not being able to tell which birds were homozygous and which were heterozygous for the dominant allele. So you wouldn't even be able to assign the genotypes. Luckily, geneticists are, well, they're not intellectually lazy. Geneticists love using their brains, but they're not so keen on manual labor or using their hands. And so they're always looking for a way to solve the experiment by thinking rather than having to spend a lot of time looking after chickens. So they want the easiest way to solve the problem. And in this case, there's a technique called the test cross that makes this kind of problem very easy. So a test cross is a special kind of mating where one parent is the parent you're interested in, and the other parent has a particular genotype. It's homozygous for recessive alleles of whichever genes you wanted to investigate. So in the case of our chickens, we want to investigate recombination in birds that are heterozygous for the C alleles and the D alleles. So we can do a test cross by mating these birds with a tester strain of chickens that's homozygous for both recessive alleles. This is extremely useful because in the offspring, we know that they always have one recessive allele of each gene, so that any time they display the dominant phenotype, for instance, a bird is black or a bird is shiny, it must have gotten that allele from this parent, not from the tester strain. So a black bird must be genotype big C, big D, little d. It must have come from a gamete from this parent that had the big C and the big D alleles. So for each of these cases, we can use the phenotype to get a direct reading, a direct inference of the gamete genotype in the bird. That's very powerful. It's also very useful that we're not confounding by having to deal with the results of crossovers in the tester strain. It's not that there's no crossovers going on. This bird probably has just as many crossovers as this bird, but in the tester strain, crossovers don't change the gamete genotypes for the alleles we're interested in because it's homozygous. So before we go on, I want to sort of nudge you about a question that beginning genetics students often kind of slide over, and that is, whose gametes was it that we wanted to examine? And the answer is we want to examine the gametes that were produced by the heterozygous F1 chickens. There's no point in looking at the gametes produced by this homozygous parent or by this homozygous parent because they'll all be the same. Similarly, we don't want to look at the gametes produced by the F2 chickens. It'll be much too complicated. These are the gametes we care about. So you do your test cross. What do you expect to see if the C and D genes are not linked? This is a critical thing to think of before you look at the results. And if you're not sure, this is an excellent time to draw a diagram or even get out little paper strips that you can write the alleles on to make sure you understand this point. And the answer is we expect to see equal frequencies of all four genotypes, all four phenotypes, and all four genotypes. You might have been tempted to pick the very familiar ratio of 3/4 of one phenotype and 1/4 of the other. This is what you'd see if you intercrossed two F1s that were heterozygous for a single gene. You might have picked the more complicated pattern because you thought, oh, more complicated, two genes must be what we expect. But this is for an F2. These are both F2s with one gene and two genes respectively. This is not an F2 population. This is a test cross. And so we're looking at the frequencies of the gametes, and the gametes will be present in equal frequencies if the genes are not linked. So here's some results. You do the test cross, and the progeny of the test cross are shown here. The first thing you want to do is just look at the numbers. Well, these are big numbers. These are little numbers. That's a pretty strong hint that in fact the genes are linked because the four phenotypes and therefore the gamete genotypes are not present in equal numbers. Here I've converted them to frequencies. Because this was a test cross, we can read off the genotype from the phenotype, and we can infer the gamete from the heterozygous parent from the genotype. The only thing left to us is to decide which of these genotypes are parental genotypes and which are recombinant genotypes, remembering that parent isn't the heterozygote, parent is the parent of this heterozygote. What alleles did they have? You could look back and see if I've given you any hints, but you don't need to because the answer is right in front of you. The genotypes that are most common are the parental genotypes, and the genotypes that are more rare are the recombinant genotypes. Of course, if the genotypes are present in equal frequencies, you can't tell, but then you know that the genes are unlinked, so it doesn't matter. Now, the final step is to use these numbers to calculate the actual recombination frequency. And here I'm asking you to calculate how far apart the C and D genes are in units called map units. And the answer is the recombination frequency is 17%, 0.08 plus 0.09. And that means that in map unit terms, the genes are 17 map units apart. So we've used a test cross to generate simple data about linkage. We started with a problem that looked like it could be pretty complicated, but once we knew we could do a test cross, it became much simpler. We recognized the evidence of linkage because the genotypes of the gametes were not equal. The genotypes of the gametes were very different. And then once we knew the frequencies of the recombinant gametes and the frequency of the parental gametes, it was easy to calculate the genetic distance separating the two genes in map units. Coming up next, we're going to talk more about genetic mapping, talking about physical maps and genetic maps and linkage in more general terms. I hope to see you there.